Welcome everybody to a Striatech hosted journal club. My name is Thomas Münch and I'm one of the founders of Striatech. I welcome you to another of our journal clubs where scientists present their projects and their data, highlighting the use and the applications of our products. Striatech is a dynamic biotech company from Tübingen in Germany. We specialize in neuroscience testing tools, in particular for vision research. The Striatech founders are all experienced neuroscientists in the field of vision research. Today, it is my pleasure to welcome Bruce Ksander as our speaker. His talk is entitled "Aging and Injured Retinal Ganglion Cells Can Be Rejuvenated by Epigenetic Reprogramming." Dr. Ksander is a professor that is associated with the Department of Ophthalmology at Harvard Medical School and with the Schepens Eye Research Institute of Mass, Eye and Ear. He received his Ph.D. in immunology from the University of Illinois, where he studied immune responses to herpes keratitis. He completed his postdoctoral training at the University of Miami Medical School and the Bascom Palmer Eye Institute, focusing on immune mechanisms regulating ocular immune privilege to corneal allografts and intraocular tumors. For over 30 years, Dr. Ksander has been conducting groundbreaking research in several key areas of ophthalmology. His work spans cellular rejuvenation of the retina and cornea, understanding the function of inflammation in glaucoma development, investigating the role of microglia in age-related macular degeneration, and pioneering the restoration of the corneal epithelium using limbal stem cells. Quite recently, Dr. Ksander has been involved in revolutionary research on reprogramming damaged cells to reverse the aging process in the eye. And I'm extremely excited to hear about this work today. Before you start, Bruce, let me remind the audience that you will be able to ask questions throughout the event and also to give a thumbs up to other questions. We will get to those questions at the end of Bruce's presentation. When you want to ask a question, the cursor by default is in the name field. Giving your name is optional, but you have to click into the question field before you start typing your question. The Q&A panel on the right will stay available for a couple of days after our live event. So if you're currently not watching live, but a recording of the event, and if Q&A is still available, please make sure to enter your question and your name so that we can get back to you with the answer. We are very much looking forward to your questions. So Bruce, please go ahead with your presentation. I invite you to unmute your microphone and to share your screen with us. And I'm very much looking forward to your presentation. You're welcome.
Okay, is that everything good with the screen?
Yep.
Okay, so I'd like to start today by acknowledging my collaborators. This work was done in a very collaborative group. 4 labs contributed to this project. My laboratory and Meredith Gregory Ksander's laboratory at the Schepens, and also David Sinclair's lab at the Department of Genetics and the Glenn Center for the Biology of Aging. I want to particularly point out Yanchen Liu, who was the single first author of this paper, and this research was his PhD thesis. He's now about to complete a postdoc in Jonathan Wiseman's lab at MIT. Also, Shigan He's lab contributed to this work, and his laboratory specializes, among other things, in optic nerve crush injury. So there are 3 types of reprogramming methods currently. Induction of IPS, induced pluripotent stem cells in vitro, the direct reprogramming of cells, which can be done in vivo, and also epigenetic reprogramming in vivo. And so obviously my talk is going to be focused on the third type of reprogramming, but just as a background to go over these different methods. So the induction of induced pluripotent stem cells was discovered by Yamanaka. The important points about this type of reprogramming is it changes the cell lineage, dedifferentiating cells back into pluripotent stem cells. It removes epigenetic signals and essentially resets the epigenetic clock back to zero. So you can do this technique in vitro, but you cannot do it in vivo. By contrast, direct reprogramming of cells can be done in vivo, and this method uses transcription factor driven lineage change. In the eye, Tom Ray has done a lot of great work in this field. He's used transcription factors to produce rods and cones from Mueller cells. There is no, I shouldn't say no epigenetic changes, but they're reduced effects on epigenetics. Importantly, though, there's no effect on the epigenetic clock. So if you look at the epigenetic age of these directly reprogrammed cells, the age stays the same. So if you reprogram an aging Mueller glial cell, it produces a rod and cone of similar age. So what we're going to focus on today is epigenetic reprogramming in vivo. And in this technique, there are no lineage changes of the cells. It also reverses the age-related CpG methylation sites, and it reverses the epigenetic clock, and obviously this can be done in vivo. So the data that I'm going to present today was from a paper published in December 2020, and all the data I'm going to talk about today is from this paper. I don't have the time to go over all of the data, and so I'm going to focus mainly on the data that was conducted in Meredith and my laboratories. So just to summarize what I'm going to present to you today is that transient OSK reprogramming in vivo resets the youthful gene expression pattern in retinal ganglion cells. So it rejuvenates cells. This coincided with restored visual function in a mouse model of glaucoma and a restored visual function lost due to physiological aging. The restoration required epigenetic reprogramming, as shown by the requirement of the DNA demethylases TET1 and TET2. OSK reprogramming turned back time on the retinal ganglion cell epigenetic clock. So since our report in 2020, 2 years later, there were several reports that also showed in vivo partial epigenetic reprogramming had a similar effect in non-ocular tissues. 2 of the papers are highlighted here. First one is by the Belmonte laboratory, who showed in vivo partial reprogramming altered age-associated molecular changes during physiological aging in mice. The other paper was from the Serrano lab. And in summary, these papers showed using various reprogramming methods, it affected a variety of tissues, muscle, skin, pancreas, liver, spleen, blood, and showed a rejuvenating effect in the methylome, transcriptome, and metabolome. And it reversed physiologic aging and also injury. And so from this now, you can see that this approach has been reproduced, and the data indicates that this is a very robust effect. Just recently, this month, a paper from the Vernier lab at McGill showed, using the same construct I'm using here, the OSK, AAV OSK, they showed protection of retinal ganglion cells in an EAE model of multiple sclerosis, and showed that this epigenetic reprogramming protected retinal ganglion cells from immune-mediated destruction. The first author on that paper is Sienna Drake, and it's in Cell Reports this month. So the reversing age by in vivo epigenetic reprogramming is derived from 4 areas of research that I'm briefly go over in the next few slides. The first is the idea that aging cells are plastic, that age can be reversed or accelerated. The discovery of a biomarker of aging, the epigenetic clock, reprogramming of cells in vitro by the Yamanaka factors, and partial reprogramming in vivo. So the idea that age could be reversed is actually not a new idea. The first evidence of this was produced almost 30 years ago by Campbell and Wilmette when they demonstrated cloning by somatic cell nuclear transfer. And so in this experiment, as shown here, the nucleus was removed from a cell and replaced. The egg cell, the nucleus was removed and replaced with a donor nucleus from an adult. And this led to the first cloned sheep, Dolly, the first cloned sheep. And so this had 2 very important dramatic effects. The first is that the factors in the egg reprogrammed this mature nucleus back into a nucleus that could support an embryo. This led to the discovery of the Yamanaka factors, which can reprogram cells and back into pluripotent stem cells. The second was that the people in the aging field said that this would not work, or a problem was that the donor nucleus was from an aged adult sheep, and because of this, Dolly would not age normally. However, it took quite a while to study a whole herd of Dolly clones, but in 2016, they studied a whole herd of cloned sheep by a variety of different assessments, and they showed that actually this was not the case. The cloned sheep all showed healthy, normal aging, indicating that what we now know is true is that the epigenetic clock in the donor nucleus, when it's reprogrammed back into a pluripotent stem cell, now has reset its epigenetic age back to 0. So this was the first real evidence that you could reset the biological clock of a cell backwards. So another important discovery in this field was by Steve Horvath and others who identified DNA methylation sites that are validated biomarker of aging and an indication of the existence of an epigenetic clock. And so this is important because this allowed us to identify the age of cells using the epigenetic markers. And this is shown here, the DNA methylation age versus the chronological age. And you can see, I think this array is from approximately 300 CPG sites. You can see there's a very, very good correlation between the accumulation of these epigenetic signals or markers and chronological age. So an important part of this was shown by Tom Ray and Steve Horvath, that the epigenetic clock actually starts during embryonic development. And in this paper in 2019, they showed using retinal development, which has a very well-known documented sequential time in the development of the different cells in the retina. They looked at the epigenetic clock of precursor cells and cells that matured in the retina and showed that they could differentiate between the different cell types in the retina during embryonic development, showing that the epigenetic clock could distinguish the age of these different developing retinal cells. So our data that I'm going to present today supports the idea that these epigenetic signals are not simply markers of age, but they actively regulate the aging process, because we've shown that when we reverse these epigenetic signals, we not only reset the epigenetic clock, but we also restore function to the cells. So reprogramming to induce pluripotent stem cells, of course, was the discovery of Yamanaka, who identified 4 genes, OCT4, SOX2, KLF4, and c-MYC, OSKM, as the reprogramming factors that you could use to take a mature cell and de-differentiate back into an induced pluripotent stem cells. And importantly, all the epigenetic signals are removed during this reprogramming, and if you block the removal of these epigenetic signals, you can't reprogram and induce iPS cells. So quickly after this discovery, people tried to reprogram cells in vitro and found quickly that it induced teratoma cells and iPSCs with totipotent features. So continually in vivo expression of OSK induces teratomas and rapid death. Malignant transformation coincides with the expression of NANOG, and so in the de-differentiation process in producing iPSCs, it's thought that NANOG expression is sort of the point of no return, where you can no longer prevent the cell from becoming a pluripotent stem cell, and in vivo, this makes the cell susceptible to tumor formation. So in the aging field, then the idea came: can you separate epigenetic reprogramming from de-differentiation? Can you remove some of the epigenetic signals without changing the cell lineage or without de-differentiating the cell? And so the first evidence of this was from Ocampo in 2016, and when he was in the lab of Belmonte, and they showed that in vivo amelioration of age-associated markers could be achieved by partial OSK reprogramming. So in these experiments, they pulsed the expression of OSK in vivo, and they showed that they were able to, using this model in a progeria mouse, increase the lifespan, and also in older mice, tissue injury models, this improved regeneration or healing of these injuries. So we became interested in the use of this model system in the eye because of the importance of aging in age-related diseases of the eye. So as we all know, aging is one of the biggest risk factors for a variety of important blinding retinal diseases, glaucoma, age-related macular degeneration, and diabetic retinopathy. So the data that I'm going to show today, we studied the OSK-mediated epigenetic reprogramming in 3 different model systems. The first was physiological aging of the retina. The other was optic nerve crush injury. And the third was the microbead-induced elevated IOP model of glaucoma. So our hypothesis for the original study was that DNA methylation clock is not just a correlate of age, but a regulator of aging. And if old tissues retain a record of youthful epigenetic information, then by partial reprogramming, you could be used to access this stored epigenetic information and reverse the age of this cell back to a younger phenotype. So Yan Chen and David knew that since c-MYC is an oncogene that negatively regulates lifespan, they hypothesized that transient expression of all 3, expression of just 3 of the 4 Yamanaka factors would be sufficient for partial reprogramming and age reversal, and that it would also be safer because it lacked the c-MYC oncogene. So the first questions we addressed were, can you get regulated expression of OSK in a single AAV2 vector, and is it safe? So the reason for this first question I'll explain in a few slides. Essentially, it was necessary to express all three of the genes within one vector in order to get the reprogramming effect. So to do this, Yan Chen developed regulated expression of a polycistronic form of OSK gene in an AAV2 virus. This is the TET on and TET off regulatory system using doxycycline. And so in our experiments, we used an intravitreal injection of AAV2, this serotype of AAV, preferentially transfects retinal ganglion cells in the innermost layer of the retina. And as you can see here, following an intravitreal injection, this flat mount of the neural retina shows the green KLF4 staining of retinal ganglion cells. And this is a cross-section showing it predominantly transfects retinal ganglion cells. So RBP-MS is a marker for retinal ganglion cells, and KLF4 is in green. And so this just shows the regulation of OSK gene expression using the TET on and TET off system. In the TET off system, OSK is expressed in the retinal ganglion cells. In the TET on system, it's off until you add doxycycline. Once you add doxycycline in the drinking water, you shut down OSK expression in the TET off system and you turn it on in the TET on system. So this is what I referred to earlier. Yan Chen found using the optic nerve crush model that if he expressed each of the three genes, OCT4, SOX2, and KLF4 in separate AAVs and combined them, injected them into the vitreous, he did not get a protective effect of retinal ganglion cells or a regenerative effect following optic nerve crush injury. However, when they were combined into a single vector, he did see the regenerative effect. And the reason for that is shown here. So this has fluorochromes of different colors associated with the different genes. You can see OCT4 is red, SOX2 is blue, and KLF4 is green. When they're all expressed in one cell, you can see here the retinal ganglion cell is white. If they express one or more of the other genes, you can see that here. When they're expressed separately, the frequency of white cells is much lower than when they're expressed in this polycistronic construct. You can see here now there's a much higher frequency of all three genes together. And so the regenerative effect of OSK expression in retinal ganglion cells depended on expressing all three of the genes within the retinal ganglion cells. So I don't have time to show all of the data, but we've done several studies showing that AAV2 OSK expressed in retinal ganglion cells and remaining on for greater than 18 months so showed no evidence of cellular dedifferentiation and no evidence of tumor formation. So I think this is important for the fact that as people who use induced pluripotent stem cells know that if you express OSK at high levels for long periods of time in vitro, you can induce iPS cells with OSK. However, the expression of OSK under an AAV is at a level which is low enough that this does not occur in vivo. So the experiments we used for the microbead model of glaucoma used this intravitreal injection of OSK, and glaucoma is an age-related neurodegenerative disease that targets retinal ganglion cells. It's the leading cause of irreversible blindness worldwide. It affects a large number of people, projected to be 112 million by 2040. There's no treatment that completely stops disease progression. So decreased visual function coincides with the loss of axons in the optic nerve of glaucomatous mice. Importantly, aging is one of the most important risk factors for glaucoma. So we wanted to determine whether AAV-OSK treatment of retinal ganglion cells restored visual function lost during glaucoma. So this is the standard microbead model to induce elevated pressure-induced glaucoma in mice. You inject microbeads into the anterior chamber of the eye and use these are magnetic microbeads, and use a magnet to draw the beads into the angle, stopping the outflow of aqueous humor. And you can see here, if you inject saline, this is the intraocular pressure on different days after injection. Saline injection, this is the baseline intraocular pressure. Injection of microbeads increases the intraocular pressure significantly for a period of a month. So using the OMR and pattern ERG measurements, we can look at the baseline measurements before we inject the microbeads, followed by 1 month of elevated intraocular pressure. We can then measure in the same mice the visual acuity and pattern ERG response to show that this elevated pressure showed or induced a decrease in visual function. Following this measurement, then we apply the AAV2 expressing OSK and express these genes for 4 weeks, and then did a final post-treatment measurement. So in this way, importantly, we're treating after we've induced a loss of visual function in these mice. And so one of the reasons we're all here is this, which is the measurement of visual acuity in mice. And so I probably don't have to go over it for this audience, but this is an example of using the optomotor reflex response to measure visual acuity. And you can see in a mouse that is in the top responding, whereas a mouse below, you can see while it is fixating, it's not responding to the pattern. And so you can measure the visual acuity by varying the width of the stripe in these mice. And so this is the results for the OMR optomotor reflex response shown here, and these are the different groups. So in the gray bar, these mice don't get microbeads, they get an injection of saline. In the other 3 groups, they all got microbead injections. 4 weeks after elevated pressure, you can see that there is a significant reduction in all 3 of these groups in the OMR response. At this time, then, is when you inject the intravitreal AAV-OSK. And so the different groups, treatment groups, are shown here. In the first group that received saline and no elevated pressure, they were left untreated. The second group received an intravitreal injection of vehicle. Then the important groups are the red and blue groups, in which they both received the OSK vector, but only in the blue group was the vector turned on. And so you can see that in the controls, there was a continued reduction in the OMR response. However, when you activate OSK in these mice here that had a reduced OMR response, there was a significant increase after 4 weeks, showing that you had rejuvenated these cells and restored the OMR response. So we also looked at the pattern ERG response. So this is stimulated by a pattern checkerboard stimulus that is a reversing pattern back and forth. It's a contrast reversing pattern, and there's no overall change in the luminescence. So it's different from the A wave and B wave ERG responses shown here. So this is the time versus the voltage. And this is a typical pattern ERG wave response. And so again, this is the same groups that I showed in the previous experiment, this showing the amplitude of the pattern ERG response. Again, in mice receiving a saline injection, this is the baseline response. After microbeads, there's an elevated pressure for 4 weeks. There's a significant reduction in the pattern ERG response. An example of these individual wave patterns shown here. This is an individual mouse at baseline and a single mouse 4 weeks post microbead injection. After, again, AAV treatment, these are the same groups that I just described in the previous experiment. Again, in the controls, the pattern ERG response remained reduced. When we turned on OSK, the previously reduced pattern ERG response was now increased. And an individual wave pattern shown here for mice at baseline, and then either following 4 weeks of elevated pressure in the controls and the control with intravitreal OSK expression, but not turned on. And then finally in the experimental group where we have now turned on OSK for 4 weeks, and there's an increase in the pattern ERG response. So the conclusions from these experiments were that OSK treatment increased retinal ganglion cell survival and restored visual function lost during glaucoma by OMR and pattern ERG, and the treatment restored function, importantly, after injury had occurred, because we documented that there was a reduction in visual function that was then restored. So we next looked at similar experiments in physiological aging. And so we wanted to determine whether OSK treatment could restore visual function lost by aging. So studies in both mice and human indicate that there is an age-related loss of visual function in mice similar to that observed in patients. So the design of the experiment is shown here. At baseline, we used 4-month-old mice as a control, and in old mice, in this experiment, we used 12-month-old mice. And so at baseline, we again measured OMR response and pattern ERG. We then injected the vector into the vitreous, as in the previous experiments, and again, either left the vector off or turned on OSK expression. Okay, then we waited 4 weeks again and looked at the post-treatment OMR and pattern ERG response. And again, so this is the baseline measurements for the young mice, 4-month-old mice, and this is the old mice. And you can see in these mice, when we inject OSK and then turn it on, there's a significant increase in the OMR response. But there is not a significant increase in the OMR response in young mice. So we next wanted to look at see whether this rejuvenation of function in cells was dependent upon epigenetic reprogramming. And so we looked at the dependency of this effect on TET1 and TET2. So these are the 1011 translocation demethyltransferases. So these TET1 and TET2 methyltransferases are essential for induction of induced pluripotent stem cells and reprogramming. So we wanted to determine whether knockdown of TET1 and TET2 could eliminate the rejuvenating effects we're seeing on retinal ganglion cells. And so to accomplish this, Yan Chen used shRNAs for TET1 and TET2. So just as a very background for epigenetics, it's a modification of DNA without changing the sequence. It can be induced by environmental effects. There's a variety of different types: DNA methylation, histone modification, and chromatin remodeling. What we're looking at here is the DNA methylation of CpG islands. And so just to show the background information for this, is that the methylated, this is a cytosine, which is then methylated. This requires a methyltransferase. The reverse of this, the demethylation of this methylated cytosine, is mediated by TET1 and TET2. And so if you look at the CpG islands in the promoter region of genes, highly methylated CpG islands repress gene expression, whereas demethylation or reduction is associated with activated gene expression. And so in these experiments, we took old 12-month-old mice, which we had showed previously that AAV-OSK injection improved the OMR and pattern ERG response. But when we injected the AAV to OSK, we included the inhibitory RNAs for TET1 and TET2. And what we found was that, as we showed before, in old mice, if you expressed OSK, you increase the OMR response. If you inject with the OSK a scrambled shRNA as a control, you had no effect, but the expression of, or the inhibition or knockdown of TET1 and TET2 was associated with a decrease in the OMR response. And similarly, if you look at the pattern ERG response in 12-month-old mice, again, there's a significant increase mediated by OSK, which is knocked down when you inhibit TET1 and TET2. So this shows that in vivo reprogramming of aging mice is dependent on TET1 and TET2, and therefore dependent on epigenetic reprogramming. And so the next experiments we performed wanted to determine, does AAV-OSK reprogramming reset the epigenetic clock? And so for this, we used anti-Thy1 antibody to enrich for retinal ganglion cells. This is the DNA methylation age in different RGC treated groups in older mice. You can see in the absence of OSK expression, this is the DNA methylation age. If you inject and treat with OSK with these scrambled controlled shRNA, you can see there's a reduction in the DNA methylation age mediated by OSK. However, if you block TET1 and TET2, you lose this significant reduction in the epigenetic clock. So then we were interested in determining what is the target of this epigenetic reprogramming in retinal ganglion cells. And so in these experiments, Thy1 positive RGCs were enriched using cell sorting from the retinas of controlled and OSK treated old mice versus young mice, 12 months versus 4-month-old mice. And then we used bulk RNA-seq to determine the relative gene expression patterns in these groups of mice. And so the data is shown in the following figures. And we first compared the up and down regulation of genes in young versus old mice. And so you can see here, so if the gene expression pattern was here, it would show that the gene level didn't change in relation to aging, whereas if the gene express went down with age, it would go to the left, up with age, it would go to the right. And then on top of that, we compared it with OSK treated and untreated cells. So again, in this axis, if OSK treatment increases gene expression, it goes up. If OSK decreases gene expression, it would go down. And so in this pattern, this axis here, shown by the dotted line, would be the pattern seen if aging is exacerbated, increased. So in other words, if a gene level went down in an aging retinal ganglion cell and OSK made it go down even further, which would be this group here, or if it went up with aging and OSK treatment made it go up even further, this axis would show age acceleration. And by contrast, the opposite axis would show a young or age reversal phenotype. So if the gene expression level in retinal ganglion cells goes down, but it goes up with OSK treatment, it would be along this axis. And so when we looked at the data, I think you can see quite quickly that the bulk RNA-seq of retinal ganglion cells is along the age reversal axis. And as you can see, so genes, the majority of genes went down with age. And you can see that the gene expression, as it goes down, the most highly downregulated genes shown here were also the most highly upregulated by OSK. And so there were 464 total genes significantly changed by aging. So all of these dots here are the genes that were significantly downregulated by aging. 268 went down, approximately 200 went up. The red and blue dots show those that were significantly changed by OSK expression. The red dots show significantly changed by OSK expression in the sensory pathway. So this is just the same data shown in another heat map type format. And you can see in young mice versus old mice, there's this significant reduction in these genes and an increase in expression in a smaller group of genes. If you look at retinal ganglion cells that are transfected with OSK, but it's not turned on, there's no change. Whereas if you turn on OSK, you now restore a more similar pattern found in young retinal ganglion cells. And so if you look at the biological processes restored by reprogramming, the top pathways are sensory perception pathways, and also, and if you just look at those sensory pathway genes, the downregulated genes that are restored by OSK expression, surprisingly, almost all of them are in the olfactory gene category, which was quite a surprise. Also affected was the inhibition of inflammation, which is consistent with the role of inflammation in aging and in glaucoma. So the final experiments I'm going to go over here are the whole genome sequencing of 780,000 CPG sites. So this data, I'm not going to show it, but showed there was a direct correlation between aging and epigenetic reprogramming. So the reprogramming induced by OSK was not global, so it didn't affect all of the CPG sites, but it was specific for CPG sites that changed with aging. And this change in CPG sites was associated with activation of polycomb receptor complex 2 and EZH2. So this is a downstream target of the OSK reprogramming, so that this is consistent with epigenetic gene regulation via activation of histone methyltransferases. So if I can just conclude now with our current working hypothesis of the mechanism for epigenetic reprogramming is that in young mice, retinal ganglion cells, there's a low level of methylation. As the retinal ganglion cells age or as they are injured, there's an increase in methylation. This impairs the function and regenerative capacity of retinal ganglion cells. If you then take these older injured retinal ganglion cells and reprogram them with OSK, OSK activates the PRC2 and EZH2, which also activates TET1 and TET2, and this results in the specific downregulation of epigenetic signals that are associated with aging. And so this then reduces the methylation level back to the young pattern. This restores function and regenerative capacity. And so the conclusions and implications of these results are that there is transient expression of OSK, which can reset the youthful gene expression patterns in retinal ganglion cells. This restored visual function occurs after injury caused by glaucoma and old age. So in other words, the dysfunctional RGCs, once they're reprogrammed, are rejuvenated and cause them to regain function. This restorative effect requires the DNA methyltransferases TET1 and TET2, and it also turned back time in the RGC epigenetic clock. And so this implies that the DNA methylation clock is not just a correlate of age, but actually helps regulate the aging process. And it also implies that old tissues do retain a faithful record of youthful epigenetic information. And so that if you reverse these epigenetic signals, the cell goes back to a younger phenotype because this information is stored. And this record can be accessed to restore and enhance tissue function and allow age reversal. So with that, I'll conclude and thank you for your attention, and I'll be happy to answer any questions.
Well, thank you, Bruce. Thank you so much. There are tons of questions, I can already tell you. That was really extremely fascinating. So let's dive right back into the questions, if you don't mind. So let's start with the first one. The first question that came in was whether this reversed aging method was only tested in mice or other models as well. Drosophila was asked, and whether it's known if this epigenetic clock is also described in other models in the first place.
The answer to the last question is yes. The epigenetic clock has been widely studied over the last 5 to 10 years, and it is a variety of different clocks have been made for all types of animals. And so there's extensive literature out there, and not only epigenetic clocks that document chronological age, but now there's clocks that document biological ages. There are also epigenetic clocks that are no longer based on CpG methylation sites, but they're actually transcriptome clocks that document age by different transcriptional patterns in cells. So there's a lot of information now on different ways to identify and quantitate biological aging. And the first question, what was the first question again?
Whether this has been tried in other models other than mouse, this reversing of aging.
So yes, it has been tried in non-human primates in a model of laser-induced neon. So it's this model was used to induce a destruction of the vessels in the optic nerve head, inducing damage to retinal ganglion cells, and it was OSK was used to rejuvenate cells following this injury. I will say I don't believe that while the rejuvenating effect has been demonstrated and a rejuvenating effect has been demonstrated, I'm not sure as yet whether it's been shown that it reversed the epigenetic clock. It may or may have not. But a variety of other laboratories outside the eye have used OSK and OSKM transient expression to rejuvenate cells and reverse the epigenetic clock.
So these outcomes are specific for RGCs because that's what's targeted by your AAV, I assume. Have any other cell types in the eye been targeted, like photoreceptors, which of course are also the target of many diseases?
So Yan Chen and I have worked on targeting RPE cells, and so we're presenting our most recent stuff at the ARVO meeting this year.
All right. So what is the duration of the OSK reprogramming effect in the glaucoma model? So
in a follow-up paper, we showed that if we expressed OSK and left the OSK on for 12 months per year, it was safe, and the significant increase in visual function was sustained for 11 months. So if you keep OSK on for long periods of time, you get a sustained increase in visual function. We have also looked at the duration if you turn OSK on for 4 weeks and then shut it off. The beneficial effect on visual function is sustained for several months and then it diminishes. So between those two experiments, I think what you can see is that we haven't yet optimized what the ideal conditions are. Both those experiments I just described were in the elevated IOP-induced glaucoma model system. And so we still have work in the glaucoma model to optimize what's the ideal treatment time to produce the longest duration effect.
What is not quite clear to me is that you lose visual acuity, visual function pattern, ERG, response strength, but these cells cannot be dead. They cannot be gone, right? Because it comes back by the rejuvenation. So what happens to them that they don't respond as well when they're still there?
Right. So I think the glaucoma field is a number of laboratories are now looking in more detail at the effect of elevated pressure. For example, the effect on mitochondrial activity, on axon transport. And during elevated pressure, all of a variety of these pathways are affected, which can affect visual function. And so it's these dysfunctional cells that are then reprogrammed and rejuvenated back to a cell type that can resist the stresses associated with elevated pressure. So I think what is unknown in glaucoma is exactly the time frame between when the retinal ganglion cells on axons are stressed and become dysfunctional, and how long do they remain in that state before they die. And so that is the window of opportunity for this type of treatment to rejuvenate dysfunctional but not dead retinal ganglion cells. And so I think in patients, there's more work to be done to see exactly what that therapeutic window of opportunity is.
Have you tried that? I forgot. In the optic nerve crush model, which should be much more extreme on the cells themselves.
So I didn't go over that data. And so what Yan-Chen showed in his experiments in optic nerve crush was that if you perform an optic nerve crush and within 24 hours express OSK and RGCs, that gene expression pattern causes regeneration of the axons. And so what role that plays in the glaucoma model, we don't know yet. We haven't really looked at axon regeneration in the glaucoma model, but in the optic nerve crush model, what he showed was that there was an increase in axon regeneration capacity.
How long does the OSK overexpression remain in the RGCs once you turn it off? And part of that,
yes.
How long does the methylation stay on?
Right. So we looked at that in a follow-up paper, and so the if I can remember the data correctly, once you remove them from doxycycline, the reduction is in a matter of weeks. It goes down to low levels.
Okay, then a question to your IOP model. So the intraocular pressure goes down again after 4 weeks or so, but the reduced visual acuity, that remains beyond the time. So that implies that the damage to the ganglion cells, or the stress to the ganglion cells, that is more permanent than the pressure itself. Is that right?
Correct. Yes. So if you look at the pattern ERG response or the OMR response following, so you elevate the pressure for 1 month and then it stops, and you then measure the visual function. We've done it for up to, I think, 5 months. It remains down, and so the reason for that is there is a loss of retinal ganglion cells and axons in this model. So again, the rejuvenation effect is preventing that loss of axons.
So you do have a window of treatment, and beyond that, it wouldn't help anymore?
Correct. So we have not done that experiment. Presumably, if you were to wait months and then reprogram with OSK, you would be past that window of opportunity and you would not be able to restore any visual function. That's a good experiment we have not done yet to find out, at least in that microbead model, what is that window of opportunity.
Now we come to the interesting questions. Is there any expectation of bringing that to the clinics and to the patient?
So there is a company, Life Biosciences, that has a license for this and is looking to bring this to the clinic.
Do you know if there are trials already?
There are not trials yet. There are trials planned in the future.
Okay. Is there anything known about the toxic effects of the OSK-AV injection in the eye, either of mice or, of course, humans looking long term?
Right. So as I mentioned, OSK has the ability to completely reprogram cells back into the pluripotent cell state in vitro. So you can use just OSK if you express at high levels for long enough. You can induce pluripotent stem cells. So the potential danger for this, obviously, is if you express the genes at a high enough level for a long enough time that you would induce dedifferentiation of cells. As I mentioned, so far, we have not seen, when we use AAV to express OSK for up to 18 months when it's on continually, we didn't see any evidence of tumor formation. But so malignant transformation is what you need to guard against in this approach.
Yeah, in your mouse model, would if tumor initiation would happen, would it happen fast enough so that he could see it in the mice?
Well, within the time frame, so yes, we looked at the morphologically and by OCT and could not detect any changes in the retinal ganglion cell, the size of retinal ganglion cells. And so histologically, they looked like normal retinal ganglion cells, and no evidence of hyperproliferation.
Mm-hmm. Then there's a question, do you know anything about lifestyle factor or diet choices that can reverse retinal aging?
No, that's not an area that I follow. That is an area out there, but I'm not an expert in that. So
it's probably an apple a day or something like that, right? So next question.
Smoking is bad for
okay.
For glaucoma and AMD, that's for sure.
Okay, next question says, Neil Peachy has a paper showing reduction in the aging mouse ERG without correlating anatomical changes. What are your thoughts regarding functional changes versus cellular changes, and how OSK expression could affect function alone?
Right. So, I mean, that's a great question, and I can tell you we want to study very carefully the correlation between cellular function and the visual function that we're seeing from this effect. So, that's what we're working on now. So I can't really answer that question other than to say that I think that's an important connection to make between what cellular pathways are affected by aging, how does OSK reprogramming reverse those pathways, and we want to know specifically what those pathways are that are affected by OSK that mediate this effect.
Does the epigenetic reprogramming have any control buttons, so to say? And how do you regulate it, that you don't get too much or too little? Number one. And number two, do you see gender differences?
So we have not seen any gender differences as of yet, but we are closely monitoring that. In the glaucoma model, we have not seen that. We are still looking at that for the retinal pigment epithelial model. So they were asking about gender differences. What else? Sorry.
How do you control that you don't get too much or too little?
Okay. So yes. So that's, I think. So clearly, AAV is a vector that is known for low levels of expression of genes for long periods of time in vivo. I think this is ideal for OSK. So, but obviously, regulating precisely the gene expression level is critical for this type of gene therapy. And I think the benefits we're seeing are largely mediated by the fact that AAV is a vector that doesn't express high levels of these three genes. Because like I said, clearly, if you use vectors in vitro and use these same three genes, you can express them at high enough levels that in vitro, they'll cause dedifferentiation of the cells. And so the safety in this approach is probably mostly attributed to the AAV in that it has this ability to express these three genes at low levels for long periods of time.
So I think I'm going to ask you one or two more questions. We're already over time, but I want to tell the audience that also the questions that we don't get to and the answers to them will eventually be posted in the Q&A section on our website. So they will be answered, if not live today. So, but maybe we can still do one or two questions.
Okay.
So one question was if there's any connection with the methylation aging clock and a circadian clock, or if these are two separate things completely.
So I am not aware of literature connecting the two, but that may be out there. That's something. Yes, I can't answer that question. It could be, but I don't know the answer to that.
And how can this data correlate with diabetes-induced vascular dysfunction?
So so the question is, could this be used in diabetic retinopathy?
Probably, yeah.
Okay, so right, so the approach for that could be to reprogram the vascular endothelial cells. We have not thought about specific targets in diabetic retinopathy, but the one approach would be to try to reprogram the microenvironment to make it more resistant to neovascularization. So that's obviously an age-related disease. So there's something associated with angiogenesis that makes the patients more susceptible as they get older. So if you find out what those age-related changes are, then you could use OSK to target those and reverse those effects so that even in aging patients, they're still resistant to those stresses.
Mm-hmm. Okay, maybe one last question. So if a cell is being reprogrammed, is it like completely reprogrammed, or like all or nothing, or is there an intermediate age that it could go to? Do you have any indication
so
of
I
that?
I think it was Wolf looked at skin fibroblasts in vitro with that in mind to look at, and they expressed OSKM at different levels to see how far you could drive the cell before you would drive it too far. And so, yes, there is an association with the epigenetic clock. We have not done it, but you could do studies to try to determine how far back you're driving the cell. But it is not. My view of it is that it's not an all or nothing response. That there's a gradation of reprogramming cells so that you can rejuvenate them a little or a lot. So I predict that it will be a quantitative effect, and how you regulate that, it's important to understand.
Okay. I suggest we leave it at that. As I mentioned, this is not all the questions that have been asked. But again, to the audience, we will post the questions and the answers in the Q&A section of the Journal Club website. Bruce, I want to thank you very much. It has been fascinating, this work, to listen to that and hearing you talk about it. I want to thank the audience for participating, for the many questions that you have asked. And I want to invite you for our next Journal Club, which will happen sometime in the early summer. And with that, I wish you a good day and goodbye.
Bye. Thank you, Thomas.